Uh, is my slide visible? Hello?
Yes, ma'am.
Can you see my slide?
Yes, ma'am,
Hello?
it's visible.
Okay. Okay. Thank you. So, good afternoon to all of you. And at the onset, I apologize for giving my talk online. I couldn't be present physically and meet all of you. Although I'm attending the CME from the very morning and hearing all the excellent talk. So, since the time is a factor, and I hope post lunch I will be able to really do some show some good data, so keep you up really. And without further, I would like to go start my talk. So if we really sorry so if we think of today, the and if we compare 20 years back, our take on cancer management, cancer therapy is far better because we have multiple new kind of therapeutic strategies like targeted therapy, immune therapy, etc., as well as good management and medical equipments. Still, we cannot conquer a single cancer, and unfortunately, therapy resistance is a major threat to cure any type of cancer. And as you all know, that the therapy resistance can be borne by the tumor cells, which could be either inherited or it could be in acquired resistance. And we also know that not only the tumor cells, but the cells surrounding the tumor in the tumor bed can help the tumor to progress to make them resistant and avoid drugs. So both the primary tumor cells as well as the tumor microenvironment are responsible for the progression of the disease and evasion of any therapeutic means. And this can evolve over time. Therefore, it is very important to understand their crosstalks, their connections, etc. So, one of the primary cancer which we are talking today is epithelial ovarian cancer, where I also work, and much not detailed because for this audience I don't have to tell about this. But the HGOCC is the preliminary, the primary subtype, which is almost 80 to 85% predominantly present all over the world as well as in India. And the therapeutic regime is also very well known to all of you, which I'm not going to talk in detail, but the recurrence is the major challenge because the cancer is often detected at late stage, and this recurrence can be classified into partially platinum sensitive relapse as well as platinum resistance relapse, depending upon the time the disease is coming back. Currently there are multiple targeted therapy, but all over what we see, even for the targeted therapy, the survival benefit is lower, and again the tumor cells can very smartly evade their therapeutic benefit and become resistant. So therefore, the whole idea or the theme of my lab is to decipher the multifacets of drug resistance, and we are actually using patient samples, neoadjuvant sample from the chemo-naive patient during surgery as well as from the relapse patient, and we have developed multiple cellular model kind of mimicking the real scenario and asking multiple questions that can we detect the acquirement of therapy resistance early, and what are the inherent signaling and biological processes that help the tumor cells to acquire resistance? Can we decipher these evolving crosstalks and the contribution of cancer stem cells? How to decode the influence of microenvironment upon resistance? And does this inherent mutational signature influence resistance? So, for today's talk, I will be the major uh, question that with these kind of answers, we can actually come up with better classification of the patient and name therapeutic strategies. And for today's talk, I will be more concentration for the last two questions, because Dr. Annapurna, whom I really forgot to thank, but she only contacted me and really invited me for this nice CME, where a lot of things on my interest are being discussed. So she only suggested to talk about the microenvironment. So, and before going to that, I also want to tell you that in my lab, we use optical reporters or the reporter genes predominantly, and these are the real-time molecular cues which comes from the marine environment or other kind of environment, and we take them. So in my lab, we use this fluorescence protein. We use the luciferase gene expression to image in the mouse, as well as when we go for human, all those kind of, although reporter genes doesn't come into the human picture, but all the imaging modalities are there. And over the years, we have developed multiple sensors so that we can bridge our understanding from live cell to living subjects. We have lots of promoter-driven reporter genes, which are basically fusion gene of luciferase and red fluorescence protein. Protein reporter will fuse luciferase with the protein, and they follow the protein level or protein expression in vivo, protein-protein interaction, as well as autophagy receptor. So coming to the theme of today's talk, that when we really talk about the cellular communications in microenvironment, this could be of two types, indirect and direct. And both these indirect and direct communications are equally important for the tumor cells, and basically a lot of signaling pathways are involved in this kind of communication. So, and these signalings could be autocrine in nature, they could be paracrine in nature, or they could be juxtacrine in nature, where the physical interaction of two different types of cells or two cancer cells are very important for this signaling pathway activation. So in my lab, we were looking at the direct interaction, which is the juxtacrine as well as paracrine. And this study basically, what I am trying to tell you about the direct interaction. So as you know, when the ascites is a predominant symptom for epithelial ovarian cancer, particularly HGOCC, and then this, due to the ascites formation, there are multiple metastases can happen, and this could be due to the passive dissemination. And so if you can see this in the peritoneum, there are small milky spots present, which are micrometastasis in a patient. And in these milky spots, there is a chance of the interaction of tumor cells as with the stromal fibroblasts, which we often call as cancer-associated fibroblasts. And what we wanted to understand, whether these interaction, how is any signaling pathway is involved, and how that signaling pathway activation actually help in progression of the disease. Now, one of the classical signaling pathway which follow this two cell interaction is the notch signaling pathway. You all might be familiar. There are multiple notch receptor, and there are ligands which are jagged and other ligands. And when they come together, there is a cleavage in the receptor happens, and this NICD basically it acts as a transcription factor. It moves to the nucleus and binds to its target gene and promote multiple biological processes. So what we did in our lab, that we developed a notch sensor, basically the notch response element driving luciferase and fluorescence protein. And if you can see, this will give you a real-time activation kinetics of the two cells interacting. So when there is a stromal cell and tumor cell expressing this particular sensor interacting, there is always the signal coming up, and based on the how much this signal is there, we can see the kinetics of the pathway activation. So we developed this model, and the very simple model. If there is no connection between the two cells, there is no signal activation. But when the two cells come into contact, there is a cleavage, and the NICD3 can act as a transcription factor and upregulate the NOX signal. To prove, we express the JAG1 in NICD3 fibroblast cells at differential level to mimic the real life scenario. And you can see when they are co-cultured, the JAG1A, which has the highest JAG1 expression as well as membranous expression, which is very important, shows the maximum bioluminescence activity, and it decreases, proportionally decreases based on the JAG1 expression, and showed the NICD cleavage as well. Then what we did, we actually collected the cancer-associated fibroblasts from aggressive high-grade serous patient sample using this magnetic sorting, and looked at their JAG1 expression. And of course, in these 5 different tabs, we find the differential activation or expression of JAG1 membranous. And when we cultured them along with our sensor tumor cell, it actually reflects the biology of the JAG1 expression and notch activation. What does it mean? What kind of the proportionate activation of the higher level of activation of notch signaling, how does it signify in the biology? We actually looked into that, and they lead to higher proliferation. This is the highest JAG expressing cells. They lead to chemo-resistance, as well as they lead to higher invasion. And then we did a target gene expression using a notch signaling pathway transcription array, and we find that here we used both the NICD3 cells, the cancer cell, as well as the CAF, and we find that P21 and VEGF-A were identified as the two most significantly upregulated gene. Now, VEGF is very important gene, and this shows that when there is a notch crosstalk between fibroblast and tumor cells, it increases VEGF and it leads to more and more angiogenesis and tumor help in the tumor growth. However, the P21 was a little bit challenging because you all know that P21 is usually senes the cells, halt the cell cycle, but there are multiple studies which show that P21 non-canonical way can actually prevent apoptosis. So when we incubated the cells with the JAG peptide, we did see high P21 expression, and when we looked at their functional implication, what we find that when we treated the cells with cisplatin, there is a significant cell death and cell death happening, and when there is a JAG1 peptide, the cell death remains going lower. So indicating that this activated notch signaling also imparting chemo resistance on the tumor cells. So therefore this juxtakinase mode of signaling, this induces higher proliferation, localized invasion through the activation of NOS3 JAG1, therefore promoting the chemo resistance and the disease progression. Now, what is happening when the cells are already resistant? Do the fibroblast cells influence, particularly for organ metastasis? Because in the juxtakinase mode, we are actually talking about the local metastasis. How it is the organ metastasis happen. So what we did, as I told earlier, that we have made cellular model which is chemosensitive, early resistant and late resistant. We injected the cells orthotopically, and we looked at the metastasis in the mouse. Very interesting, as the cells becoming resistant, they show higher and higher metastatic propensity into different organs, and lung is one of them, where we see 80% of the lung showed metastatic nodules by the late resistant cells. They invaded there. So what we did, we collected, isolated the mouse lung fibroblast with many other observations, which I'm not going into detail. We showed that this lung fibroblast in presence of the Sorry. So we basically see that it secretes IGF-1 and that leads to proliferation invasion. Now, interestingly, what we did a subtracted LCMS and find that S104 is secreted by the cancer cells when they are cultured in the FCM, the lung fibroblast cells, and the secretion is much higher in the lead resistant cells. What does it mean? This S100K4, you know, it's a calcium binding protein having role in multiple metastatic processes, and we downregulated the S100. We did see lesser secretion, and when we injected these cells in the mice, the lung metastasis is absolutely abolished. We next find that there is a small molecule niclosamide, which is an anti-emetic drug. It can bind and inactivate S100R4. So when we injected the mice with niclosamide, again the same very important result had come, that it actually taken care of all the metastasis, particularly in the lung. So all this showing that how the microenvironment in physical contact or in a distant mode, it can actually influence the resistance and metastasis. But as you know that these HGOCC are very complex genomic state, they have high intratumor heterogeneity and chromosomal instability. So we also are trying to understand if the tumor cell is having inherently genomic heterogeneity, how does it interact with the microenvironment. So this is TCGA data known to all that P53 mutation is the major characteristics of HGOSOC, and this also doesn't change in the relapsed set of patients, the same P53 mutation retained. So what we did, we collected the ascites from multiple patients based on they could be the sensitive chemo-naive or they are platinum resistant or platinum sensitive. We looked at their platinum sensitivity. Very interestingly, the patients who are platinum resistant, they showed much higher IC50 value compared to the chemo-naive or the platinum sensitive patients. And we did the targeted genomic sequencing. This is probably the first genomic sequencing in Indian ovarian cancer patient of the relapsed patient, including the relapsed patient. We did see much higher somatic BRCA2 mutation compared to the West population and multiple P53 mutation. Now we are focusing on majorly mutant P53 because you know they are very different in nature, exerting their function than the wild type P53. So we created a library and we find that different mutation give different kind of resistance status to the cell towards platinum, and they are also showing different kind of tumorigenic behavior, like this R282W, which is very highly resistant, show highest migration capability, highest invasion capability compared to the other and compared to the wild type P53. So this study is ongoing. I'm kind of really rapidly filing because I know the time is a constraint. So overall, what I'm trying to tell you that yes, the microenvironment has a significant influence, which you are probably also seeing there. If there is a physical contact and not JAG1 signaling is on, that leads to chemoresistance, proliferation, invasion, as well as angiogenesis. When there is a paracrine mode up there, it also helps in organotropic metastasis and these tumor cells with different P53 mutation. Now currently we are evaluating that how they will influence or the construct is different than with the CAF, and that is affecting the disease progression. So this coming to the end of this my talk. I'd like to thank all my students. I predominantly talked about Abhilash and Shobik and Megha and Saurabh's studies. Without them, these beautiful studies would not be possible. And I have all the funding agency. I thank you and happy to take any question. Am I within time or did I exceed the
Thank you so much for being on time. And I'm Dr. Annapurna here. Dr. Preeta, I just wanted to ask one question.
Yes.
Any questions? First, I'll ask from the audience. Any questions from the audience? Okay, my question is with respect to the heart signaling pathway, did you also study the hormones associated or role of hormones and HGSOC?
No. We haven't looked at the hormonal influence of this on this particular pathway. It's an interesting question, actually.
And one more thing, I mean, with respect to the P53 mutations that you were telling, and did you also do the, could you find a difference in the NGS also? With respect
Yes, so we actually identified two P53 mutation which have not been reported in Western population or any other HGOCC. So these two are novel P53 mutation, and I haven't talked on other studies, but we actually are doing this study that particular mutation make the cells more sensitive to a therapy, which is the alpelisib, a PIK3CA inhibitor. So definitely different P53 have their role, and we identified two new P53 mutation from our Indian cohort.
So not all P53 mutations are same. I mean, they are all different, even genetically and behavior-wise. Even behavior also, functionally also they are different.
Yes. And so that is coming to another, actually putting another complexity that when we treat the patient, maybe a clone of P53 cells or tumor cells which is harboring a particular P53 mutation may be more resistant, they will not be eliminated, and they will behave differently along with the microenvironment and lead to a different kind of disease progression or probably different kind of organotropic metastasis as well. So this is a huge field, even though we are studying P53, but we really don't know much about it. So that's one area.
Thank you so much.
Is there any other question? And I really hurriedly said, and
thank you, Dr. Preeta.